Hi guys, I'm Julie.
Today, we're going to teach you about a very important anaerobic process called
Lactic acid fermentation. Do you know what that means? Well, if you answer no, don't you worry because we're going to help you out. Okay, so this is a process of glycolysis, which is the first step during lactic acid fermentation. And it happens in cellular respiration as well because it does not require any oxygen. So we begin glycolysis with the glucose molecule, which is a 6-carbon molecule, as you can see in this picture. And the first thing we have to do is add a phosphate group from an ATP molecule to the glucose molecule twice. So we end up with this fructose 1,6-biphosphate, which basically means that you have a phosphate molecule added to the first and the 6th carbon of the glucose molecule.
Yeah. And this happens because in order for glycolysis to occur, an input of energy is required. So once you have the fructose 1,6-biphosphate, the molecule then splits, starting the second half of glycolysis and production of pyruvate.
So after this molecule splits, we have 2 pGol molecules, and as you can see, each one of them has 3 carbons and 1 phosphate. So what happens here is this NAD plus molecule, which is very important in this process, is reduced by accepting a hydrogen from this molecule, becoming NADH, and a phosphate in this process is added to the pGol molecule so that now you have 3 carbons and 2 phosphates in this molecule. And that happens twice because we have 2 of those. So as we know, your main aim in glycolysis is to create ATP molecules, which is like the energy currency in your body, and that's what your cells need in order to function. So what happens here is you have ATP molecules which have 2 phosphates, and they're going to take a phosphate from the pGol molecule and become ATP from there. So you have 2 of them, because since you have 2 phosphates, you need 2 ATP molecules coming in here to become 2 ATP molecules, and then you end up with this 3-carbon molecule with no phosphates, which is the pyruvate. And once again, you have this happening twice. Once glycolysis is over, given we have no oxygen when we're speaking about lactic acid fermentation, our cells have no oxygen to function as a electron acceptor in the electron transport chain, as we said before. So this is why lactic acid fermentation is different than cellular respiration, because after this, for cellular respiration, you would be going into the Krebs cycle, but instead, we're just going into the lactic acid fermentation, which is when you're going to be producing lactate, and we're going to see this happening in the next slide.
In summary, from glucose goes through glycolysis, and the slide shows that there's a result of 2 ATP molecules, and even though the previous slide we showed 4 ATP molecules were reduced, there are also 2 ATP molecules used in the beginning of glycolysis as the investment to begin the process. So that is why there are 2 ATP molecules following glycolysis.
And as we said before, glycolysis ends up with, after glycolysis, you have 2 pyruvate molecules, which is your end result, basically. And the pyruvate is what will be now the reactant in the anaerobic process of lactic acid fermentation. So basically what happens during lactic acid fermentation is you have those 2 NADHs that were produced during glycolysis come in and donate 2 hydrogens into the pyruvate molecules and produce 2 NAD pluses. So it's basically being oxidized now because it's donating the hydrogen to the pyruvate, and the pyruvate is being reduced. So after lactic acid fermentation is over, you end up with 2 lactate molecules, and that's your end product. So now, just for us to better understand this, let's follow the hydrogen atoms throughout this process, right? So after glycolysis, you end up with 2 pyruvate molecules, and each one of them has 3 hydrogen atoms, right? And then as they go through the lactic acid fermentation process and the enzyme lactic acid dehydrogenase comes in to help out. You have the two NADH molecules that were produced during glycolysis come in, they donate these 2 hydrogens to each pyruvate molecules, and then you end up with a lactate molecule that has 5 hydrogens, which makes sense because it's 3 plus 2 coming in from an NADH. So basically, this means that glycolysis, the purpose of glycolysis, besides producing ATP, is to produce NADH, which is then used during lactic acid fermentation to produce NAD+.
And NAD+ is recycled back into the glycolysis process to again produce 2 ATP and the pyruvate molecules.
So if we go back to the picture here, we see that NAD+, which was regenerated during lactic acid fermentation, is used here during glycolysis to produce the NADH. So it's like a cycle. You need the NADH from glycolysis to go through lactic acid fermentation, and you need the NAD+ that is produced during lactic acid fermentation to be used in glycolysis. And this is why we have lactic acid fermentation happening when you don't have oxygen to go through the electron transport chain, because you still need to produce ATP. So how do you do that? Well, you have to rely solely on glycolysis to happen. So you have to keep producing the NAD+ that can be used in glycolysis over and over again. And that's why we have lactic acid fermentation.
I said, oh my, what a marvelous tune. It was the best night, never would forget how we moved. The whole place was dressed to the nines, and we were dancing, dancing like we're made of starlight, like we're made of starlight.
This is the intense soccer. Oh, it's starting to burn, you guys. Whoa, what's wrong with my leg? Oh my God, it's burning. Wait, mine too. Oh, mine too. It's burning. It's burning. It's burning.
Intense exercise, the blood pH lowers because of lactic acid buildup, causing tissue in the muscle cell to have lower pH. And this is what studies suggest show. Studies suggest this is what causes the burning sensation.
Okay guys, so just a quick summary of what we spoke about today was lactic acid fermentation, which begins with glycolysis, a process that also occurs in cellular respiration because it does not require oxygen. And the second thing that happens is when we have our 2 pyruvate molecules, they go into the process called lactic acid fermentation, where they receive hydrogen from the NADH molecules produced during glycolysis, regenerate NAD+ molecules, and are basically transformed into lactate molecules. So why does lactic acid fermentation happen? Well, because we have no oxygen to function as the last electron acceptor in the electron transport chain. And what's the purpose of lactic acid fermentation? Well, the purpose is to regenerate NAD+ so that it can be used over and over again during glycolysis in order to produce the 2 ATP molecules that are the result of glycolysis, as we showed you before.
For the body for energy. And the body needs it for energy.
So, and our final thing is the connection to the real world, which is that lactate causes the burning sensation that the soccer girls feel after a very intense soccer game. Okay, thank you very much for watching our videos. We really hope this was helpful, and good luck on your AP Bio classes.
And if you want more definitions and an explanation and summary of lactic acid fermentation,
Click below!